introduction. I'm excited to share with you all what I've been working on for the last 4 or so years and introduce you a little bit to the work that I'm starting in my own lab, all of it aimed at understanding how feeding circuit activity is regulated by in vivo nutrient sensing in the gut, and how these dynamics change in a variety of disease states, in the case of today's talk, focusing on obesity. So, sorry, this is, okay. So after a brief overview of energy homeostasis and specifically the role of AGRP neurons in appetite regulation, I'm going to be telling you a little bit about how these neurons respond to nutritionally relevant stimuli that originate in the gastrointestinal tract, namely gastric nutrients or food that you've just eaten and satiety hormones that are released in response to the food that you've just eaten. And then finally, I'm going to discuss more recent findings detailing how these dynamics change in response to diet-induced obesity. I'm going to show you that diet-induced obesity leads to nutrient-specific changes in the dynamics of AGRP neurons, which are incompletely reversible, and suggest a possible circuit-based mechanism for weight regain following weight loss in obese people. So very briefly, energy homeostasis is a really stunningly robust process that works incredibly well. And if you think about it, it actually results in the ability of billions of people to maintain their body weight and metabolic health in this really delicious world without struggle or without even really much conscious thought. This, of course, occurs by balancing food intake and energy expenditure. And when you look at older longitudinal studies, nearly 50% of people are able to maintain this energy balance within about 6 kilocalories per day over the course of 10. Put that into perspective, this is a margin of error of about a ninth of an Oreo cookie, about a quarter of a cup of broccoli, or a little bit less than a teaspoonful of macaroni and cheese. So this is very clearly a balance that's far more precise than even a very avid calorie counter could do consciously. And not surprisingly, accomplishing this is extraordinarily complex. And accordingly, the study of energy homeostasis encompasses a broad range of fields involving nearly every organ system. And what I study is, and what I'm going to tell you about today, is a piece of this that focuses on the interaction between 2 organ systems, specifically the gastrointestinal tract and the brain, communicating about recently consumed nutrients. This communication allows an animal to appropriately drive behavior, for example, telling an animal to stop eating, and also helps to control metabolism. And I'm going to give you a brief and oversimplified view of how it works here. So when you eat, you wind up with a lot of signals in your gut. Initially, you have the nutrients you've just consumed and their metabolites, usually some combination of carbohydrates, lipids, and peptides and amino acids. And this results in the release of a large and growing list of satiation factors, many of which come from these specialized enteroendocrine cells lining the intestinal tract, and some of which come from other gastrointestinal organs, namely the pancreas. And the goal of all of these signals in your gut is, as I mentioned, to modulate your behavior. And in order for this to happen, of course, these signals from your gut need to somehow be communicated to your brain. And this happens in one of a couple of ways that are not mutually exclusive and likely both important towards the regulation of energy balance. First, some of these signals, metabolites and hormones, released from enteroendocrine cells can act locally at vagal afferent terminals, which richly innervate the entire gastrointestinal tract. These pass through these neurons have their cell bodies in the nodose ganglion and then synapse in the NTS in the brainstem, from where they project broadly throughout the brain. In addition, these signals, both the metabolites and the hormones, as their function implies, wind up in your circulatory system, and some of them may act directly on the brain, either by crossing the blood-brain barrier or by acting at circumventricular regions that lie outside the blood-brain barrier. And one really important circumventricular brain nucleus involved in mediating appetite is the arcuate nucleus in the hypothalamus. This nucleus houses a critical population of neurons called AGRP neurons, which are essential for promoting hunger. And the function of these neurons will be the subject of the rest of my talk. Now, conveniently, this tiny population of neurons, shown here in green, about 1,000 cells, is uniquely genetically defined by the neuropeptide AGRP, and that renders these neurons genetically tractable. And in fact, the selective genetic manipulation of these cells has been used for well over a decade now, and has been used to really prove definitively that these neurons are critical for regulating feeding in vivo. So in the mid-2000s, Richard Palmiter's group used selective diphtheria toxin mediated ablation of these cells to show that this population of neurons in adult mice is absolutely necessary for driving feeding. So when you express diphtheria toxin receptor under the control of AGRP, then treat with diphtheria toxin, these cells die. And as you can see in red here, over the course of about 5 days, an animal's food intake plummets until you have to sacrifice them after about 5 days. And in fact, it's not just that these mice won't eat, they will actually reject food that you gavage in them. They have no interest in food at all. A bit later, with the advent of optogenetics, it was shown that stimulation of these cells is actually sufficient to drive feeding by Scott Sternsen's group and others, and that's depicted here. In a sated animal, if you optogenetically activate AGRP neurons, you see the induction of robust feeding compared to a control animal that doesn't express channel rhodopsin. So AGRP neurons are necessary and sufficient for feeding, but these while these and similar studies definitively show that AGRP neurons are required for feeding, these are all pretty blunt, non-physiologic manipulations of the activity of these cells. And it wasn't until calcium imaging became more widely available that the question of what these cells are doing in vivo could be addressed more directly. And based on the studies I just showed you, in addition to a lot of prior work on AGRP neurons using combinations of slice electrophysiology, behavioral pharmacology, histology, and other ex vivo methods, people kind of had a model of AGRP neuron function that looked something like this, where we predicted that their activity would gradually rise as an animal fasted over several hours. And then once an animal started eating, nutrients would be absorbed from its gut, hormones would be released, and over the course of about 10 or 20 minutes, as the AGRP neurons sensed these changes, we predicted that AGRP neuron activity would gradually decrease over the course of many minutes. But this is not at all what was found during the first calcium imaging recording from AGRP neurons, which was done by Zach Knight's group. And what was found really changed the way the field views these neurons. So shown here, non-food objects, as you can see, had no effect on AGRP neuron activity. But when a hungry mouse is presented with food, rather than seeing gradual inhibition over many minutes of AGRP neurons, you instead see that AGRP neuron activity is inhibited over seconds upon the initial presentation of food. And in fact, this inhibition is nearly complete by the time the animal takes even its first bite of food. So this inhibition is modified by a bunch of different factors. First, it's modified by an animal's nutritional state. So shown here in red is the response to chow presentation seen in a fasted animal. So a hungry animal has this nice big drop in AGRP neuron activity when you present chow. By contrast, if you present a sated animal with chow, it'll have a much smaller response to the food. This inhibition is also modified by the palatability of the food that's being presented. So a really delicious food like peanut butter will cause a greater AGRP neuron inhibition than a less delicious food like chow. And taken together, this led us to kind of have the hypothesis that the magnitude of AGRP neuron inhibition predicts imminent food consumption. And I tested this hypothesis in a few different ways when I joined Zach's lab and found that it holds true really across a broad range of conditions. So each of these points represents a treatment or manipulation that alters the appetite of a group of mice. And we recorded from the AGRP neurons of these mice after these interventions and presented them with chow. And on the x-axis, you see the magnitude of the inhibition of AGRP neurons they showed when I subsequently presented them with chow. And then on the y-axis, you see the amount of food they consumed over the ensuing 20 minutes. And you can see that there's a really pretty robust relationship between these two factors. And my favorite example of how basically an animal's internal state and hunger modulates the magnitude of anticipatory AGRP neuron inhibition and how faithfully this predicts food intake is LPS. So many of you probably know that LPS, or lipopolysaccharide, is a component of gram-negative bacterial cell walls, and its administration mimics severe infection in mice, causing really profound malaise and anorexia about 4 hours after administration. So for this experiment, mice were overnight fasted and then given either LPS or saline. 4 hours later, as they developed malaise, we started recording from their AGRP neurons, and then subsequently presented them with chow. And you can see that animals that receive saline, they've now been fasted for about 18 hours overnight, plus the 4 hours after their injection. These are very, very hungry animals, and they have a really nice, robust response to the presentation of chow. By contrast, animals that have been pre-treated with LPS have a much attenuated response to chow presentation. And we noticed during this experiment that the animals kind of neatly divided into two groups. Most animals don't eat at all 4 hours after LPS administration, but some animals continue to have a little bit of an appetite. And post hoc, if we divide the mice into these two groups, you see that the vehicle results in this nice inhibition that I showed you. Mice that do eat a little bit after LPS administration have this intermediate response, and then mice that don't eat at all really truly have no response of their AGRP neurons. So the response of their AGRP neurons to the presentation of chow really faithfully predicts how much they're about to eat across these conditions. And here's a similar graph to what I showed you earlier. Here's the magnitude of AGRP neuron inhibition on the x-axis, and subsequent food intake on the y-axis. So this was all, as I mentioned, incredibly surprising. And I think the big takeaway from these early calcium imaging recordings from AGRP neurons is that the primary in vivo mediator of AGRP neuron dynamics is actually not nutrients, not nutrients in the internal environment, but rather sensory cues in the external environment, which cause rapid inhibition that predicts future food intake. But it seemed almost implausible to us that these neurons, which are really critical for preventing starvation, would irrevocably shut off when you smell or see food, because you can imagine a lot of things that might prevent you from eating that food. And if cues eliminate your drive to eat, you will starve. Moreover, there had been 20 years of research that really pretty directly connected satiation and satiating hormones to the activity of AGRP neurons. And so I wanted to tease apart what the actual role of nutritional signals were in regulating AGRP neurons. And as a first test of this, I did an experiment with what we call caged chocolate. So again, starting with a fasted mouse, we record from AGRP neurons and initially present this mouse with what we call caged chocolate. This is a piece of chocolate underneath a wire basket that the mouse can see and smell, but it cannot eat. After 20 minutes, this caged chocolate is removed, and then subsequently accessible chocolate, or chocolate the mouse can see, smell, and eat, is presented. And what we found is that when animals are presented with caged chocolate, you see this immediate drop in AGRP neurons, which we had predicted would happen, because that's what happens when a mouse sees or smells food. But unlike when animals are actually able to eat following food presentation, the activity of AGRP neurons over a period of about 10 minutes actually returns to near baseline in these animals presented with caged chocolate. By contrast, when we give the animal available chocolate 20 minutes later, it has this robust AGRP neuron inhibition, which then is sustained for 20 minutes while the animal eats, and is also sustained for at least 20 minutes following removal of the food. So from this experiment, we concluded that food intake is necessary to stabilize the rapid sensory inhibition of AGRP neurons. And what I was really interested in studying from here, what are the nutritional factors that are that are required for this process. But unfortunately, this is not something I could study in freely feeding animals because as soon as an animal sees or smells food, you basically lose the entire dynamic range of activity of these AGRP neurons. And so to circumvent this, this problem, I developed a model to avoid the sensory stimuli that are associated with normal eating, using intragastric feeding during fiber photometry recording from AGRP neurons. So basically, these mice are prepared with a chronic intragastric catheter through which we can infuse whatever nutrients we want at whatever rate we want while recording from AGRP neurons. This is a version 1.0 catheter. We have much smaller, tidier catheters now. And basically what we found is that infusion of nutrients directly into the stomach of these mice was sufficiently to rapidly, this time within minutes, and durably inhibit AGRP neurons. So this is shown here for glucose, a lipid mixture, and peptide mixture. We found similar results when we infused a large variety of different mono and disaccharides. So the inhibition is independent of the macronutrient identity we're infusing. And we found that it's proportional to the number of calories we're infusing. So within any given nutrient that we infuse, the magnitude of the inhibition we find over the course of infusion correlates well with the concentration of the nutrient we're infusing. Not shown on this graph, but non-nutritive infusates do not inhibit AGRP neurons. Specifically, non-caloric sweeteners, we used sucralose, show no effect on AGRP neuron activity. We next wanted to begin to dissect how nutrients in the gut accomplish this inhibition. So I showed you a list earlier of a large number of enteroendocrine cell-secreted factors that drive satiety. And the strategy that we took initially to dissect the mechanism of nutrient-mediated AGRP neuron inhibition was essentially to screen through a large number of those hormones and see whether any were sufficient to inhibit AGRP neurons when administered peripherally. And while many of these factors did absolutely nothing to AGRP neuron activity, we found that a subset of these, the hormones cholecystokinin, peptide YY, and serotonin, were sufficient to inhibit AGRP neurons on a time scale roughly consistent with fast refeeding. So shown here on the left is the response of AGRP neuron activity to CCK administration, just a peripheral intraperitoneal injection of CCK, compared to a PBS vehicle, which has no effect on AGRP neuron activity. As I mentioned, PYY and serotonin are also sufficient to inhibit AGRP neurons. Some notable hormones which did not affect AGRP neurons, at least not on this time scale, were leptin, GLP-1, and oxyntomodulin. So we had now a list of a few hormones that are sufficient to inhibit AGRP neurons, but and that's all well and good. But the question we're really interested in is not whether we can inhibit AGRP neurons by giving a pharmacologic dose of a hormone. What we really want to know is whether these hormones are actually involved in nutrient-mediated gut-brain communication. In other words, are they necessary? And so we approached this using a pharmacologic experiment, which is described here. So mice were habituated to a recording chamber, and then a receptor antagonist to one of these hormones that led to AGRP neuron inhibition was administered, or a vehicle. After roughly 5 minutes, either water or nutrient was infused. And I'm going to show you here the results using a CCK receptor antagonist. Here in black, you see what happens with water infusion, which is nothing, and the pre-injection of a CCK receptor antagonist really does nothing to the baseline signal of these neurons. Then in red here, you see what happens when we infuse a vehicle, and then followed by a lipid infusion. So you see this robust lipid-mediated AGRP neuron inhibition, similar to what I showed you previously. But when we pre-treat with a CCK receptor antagonist, you see that the subsequent response to lipid is almost completely abrogated. Interestingly, when we repeated the exact same experiment using a glucose infusion instead of lipid, we found exactly the opposite result. So again, water, there's no effect. In gold here is the response to glucose after pretreatment with vehicle. And when we pretreat with a CCK receptor antagonist, we show absolutely no effect on AGRP neuron activity. So from this experiment, we concluded that the peptide hormones cholecystokinin is necessary for lipid-mediated AGRP neuron inhibition, but is totally dispensable for glucose-mediated AGRP neuron inhibition. And thus, while all macronutrients cause AGRP neuron inhibition regardless of their identity, the mechanisms by which each macronutrient accomplishes this appear to be nutrient-specific, and the mechanisms for both carbohydrate and peptide-mediated AGRP neuron inhibition actually remain unknown. And I should also add here that we, as I mentioned, both serotonin and peptide YY are sufficient to inhibit AGRP neuron activity over similar timeframes. And we did try using a pharmacologic approach to determine which of those are necessary for nutrient-mediated AGRP neuron inhibition. But both of those hormones have multiple receptors, and that really complicated interpretation of those experiments. So the roles of those two peptides in AGRP neuron inhibition remain uncertain. So basically, to recap quickly this part of my talk, over the last 5 years, we've gone from a model of AGRP neurons as more or less passive detectors of nutritional state, whose activity increases gradually with fasting and decreases gradually as the nutrient composition of our blood changes following feeding, to viewing them more as active integrators of nutritionally relevant data across multiple timescales. So these neurons integrate within seconds exterosensory cues that predict food availability. They integrate within minutes signals from the gastrointestinal tract about recently consumed nutrients. And then some data, which I didn't show you today, their basal activity is actually affected over hours to days by leptin and possibly other hormones that track longer-term nutritional state. So this information together is used to drive appropriate behavioral and metabolic output. And moving forward, my primary interest was basically to begin to understand how these more rapid dynamics, so the dynamics over seconds and over minutes, were altered in obesity. So basically, having spent the first part of my presentation deconstructing some of the mechanisms of energy homeostasis, which I highlighted as being this really robust system that works for many, many people, it's also pretty easy to see that in our current environment, the system really fails a lot of people. And in addition to weight gain and the initial development of obesity, a huge part of the obesity epidemic is that patients with obesity have an incredibly hard time maintaining weight loss. This has been illustrated over and over again, perhaps nowhere as depressingly as on the really awful reality TV show, The Biggest Loser, where severely obese people lose weight through dramatic lifestyle interventions on TV for your entertainment. And in some remarkable studies by Kevin Hall's group, contestants were followed for over 6 years, starting before and going for 6 years after their appearance on this TV show, and they had a large number of metabolic parameters monitored during that time. But one of the most striking things to me about these studies is that nearly all contestants regained the majority of the weight that they lost. So that's shown here in red. So they lost something like 60 kilograms, which is really, really a remarkable amount of weight to lose through lifestyle interventions. But by 6-year follow-up, they had regained at least 40 of these kilograms on average. Similar results have been observed in other clinical studies, and also, I'm sure, anecdotally in many of our lives, either our own or family or friends. But the mechanisms underlying these changes, particularly at the level of appetite control, remain pretty incompletely understood. And so I set out to understand how AGRP neuron activity specifically in response to nutritionally relevant stimuli changes in obese mice. And to do this, I used a high-fat diet-induced model of obesity in mice that were equipped for fiber photometry recording from AGRP neurons. I used, for those of you who are interested, a 60% high-fat diet. So this is a very, very high-fat diet, and focused on the responses of AGRP neurons to the sensory detection of food in the environment, to intragastric nutrients, and specifically to the hormone cholecystokinin. I basically recorded the response of mice to these stimuli at baseline, following 6 weeks of a high-fat diet, and then again 4 weeks after a return to a chow diet, after which an animal had lost some weight. And a separate cohort of mice that were maintained on chow were recorded at these same time intervals to control for any loss in calcium imaging signal. And shown here are their body weights over this time course. You see the control mice maintain relatively stable weight over this time, whereas the diet-induced obese mice exhibit pretty significant weight gain over about 6 weeks, and then partial weight loss following a return to chow, which I'll come back to in just a moment. So I started with really the simplest experiment, asking what happens to the anticipatory response of AGRP neurons to food cues in obese mice. And we found that it was strikingly impaired with weight gain. So here's a baseline response of AGRP neurons to chow detection in black. And after 6 weeks on a high-fat diet, you see the response severely attenuated in red. This significantly, but not completely, recovered following weight loss. And you can see control mice did not exhibit this pattern. And this is quantified here. This experiment that I've shown on the screen is the response with chow presentation. We had a concern that basically giving an obese mouse that had been on high-fat diet for 6 weeks a chow pellet might be a little bit like giving a young kid that's been raised on pizza a salad to eat. And so we repeated this experiment, presenting the mouse with high-fat diet instead of chow, and showed a similar pattern when mice are presented with high-fat diet. And so I told you just a few moments ago that in normal weight mice, the magnitude of sensory inhibition correlates well with imminent food intake. And it turns out that in obese mice, this correlation is really faithfully maintained. So after 6 weeks on a high-fat diet, you see this dramatically attenuated sensory response to chow, and in association with that, you see a dramatically decreased amount of fast refeeding, and this is on chow. This had actually been reported in the literature before. And then following recovery or weight loss on a normal chow diet, you see a return to more normal AGRP neuron dynamics, and actually a return completely to baseline of chow consumption here. And interestingly, this return to totally normal chow, to totally baseline chow consumption occurs even though the animal has lost only a fraction of the weight that it's gained. So this mouse is still significantly obese compared to its baseline self, but it's eating just as if it were at its baseline body weight. And overall, we think that the decreased so I guess taken together, we believe that the decreased dynamics and fast refeeding in obese mice may actually represent accurate tracking by AGRP neurons of energy you need in the obese state. These fat mice just aren't getting super hungry after an overnight fast because they're in this chronic positive energy balance state. Moreover, the rapid return to normal fast refeeding before the weight has fully returned to baseline may actually be a marker of an altered set point in these mice. In other words, as soon as they begin to lose weight, their brain, which now believes they belong at this new higher body weight, senses the negative energy balance and thinks that they're starving. And so overall, we think that the AGRP neurons are accurately tracking energy need through weight gain and weight loss. So to better understand this phenomenon of decreased AGRP neuron dynamics and decreased fast refeeding in obese mice, and also to clarify the extent to which this hypothalamic circuit even drives feeding in obese mice, we used an optogenetic approach. And I showed you before that AGRP neuron stimulation is sufficient to drive feeding in sated animals. But we now know that this model is actually pretty non-physiologic of stimulating AGRP neurons as animals eat voraciously. And we know it's non-physiologic from the calcium imaging work that was done by Zach Knight's group and has since been repeated by many others. And basically, what we've learned about AGRP neuron activity is that when an animal is actually eating, these neurons are turned off. So AGRP neuron activity is really high when a mouse is hungry, but drops as soon as it sees food before it starts eating. Whereas here, feeding is being driven by actual activation of AGRP neurons. So a student in Zach's lab developed a novel optogenetic stimulation paradigm to address this, known as pre-stimulation. And it's pretty simple. You have a mouse in a cage without access to food, and you turn the laser on to stimulate their AGRP neurons in the absence of food. You then turn the laser off and present the food to mice only after the laser is turned off. And what Yiming and Zach's lab found is that this results in voracious feeding in sated animals, similar to what was seen in the more traditional AGRP neuron stimulation paradigms, where mice eat voraciously for up to after 60 minutes of pre-stimulation, they eat voraciously for up to at least 60 minutes after food presentation. And this feeding is actually comparable to the amount seen here in the more classic stimulation paradigms. Subsequent work by Mike Krash's group has shown that pre-stimulation is actually a weaker driver of food intake in more natural situations where competing interests are present. So where competing interests are present, co-stimulation, as seen here, will drive more feeding than pre-stimulation, as shown here. And I basically used both of these paradigms to assess the role of AGRP neuron activity in feeding in obese mice. And our experimental design is shown here. Mice were feeder trained and then received 3 optogenetic stimulation sessions at baseline, which were repeated again after, in this case, 4 weeks of high-fat diet-induced obesity. So at each of these time points, mice had a no stimulation control trial where the laser was off during habituation and during food availability. They had a pre-stim trial where they received AGRP neuron stimulation prior to food availability, and then a co-stimulation trial where they received laser stimulation during food availability. And what we found is that while at baseline, as Yiming had shown previously, pre-stimulation in light blue and co-stimulation in dark blue cause comparable food intake. In obese mice, pre-stimulation actually led to dramatically attenuated food intake compared to co-stimulation. And we saw a similar pattern when the food delivered was low-fat pellets, grain pellets, versus high-fat diet. And so taken together, I think that these, we drew basically a couple of different conclusions. First of all, basically stronger AGRP neuron inhibition is required in obese mice to stimulate comparable feeding. And we think that this means either that there are downstream impairments in the circuitry, which may be responsible for blunted refeeding observed in obese mice, and also other circuits may be involved in driving normal feeding in these mice. So we've shown that obesity blunts the anticipatory dynamics of AGRP neurons, but that these dynamics still faithfully track food intake and that they're substantially reversed by weight loss. And further, we believe this to be an adaptive response to sustained positive energy balance in these mice. And we've shown that AGRP stimulation leads to decreased food intake in these animals, suggesting downstream impairments and possible parallel circuits being involved in the process. But what happens to the gut-mediated signals in these animals? So we used a parallel approach to address this question. Mice that were equipped for intragastric feeding, as well as AGRP neuron photometry recordings, underwent the same high-fat diet-induced obesity protocol and weight loss. And what we found is that diet-induced obesity selectively blunts the response of AGRP neurons to dietary fat. So at baseline, we see this nice fat response in black, as I've shown you previously, and there's a modest but significant inhibition of this response in diet-induced obese animals. By comparison, the response of these mice to intragastric glucose delivery and intragastric peptide delivery remain unchanged. Strikingly, unlike the response to chow presentation, the attenuated response to intragastric lipid persists even after weight loss. So here in the diet-induced obese animals, you see after 4 weeks of weight loss, you can't really see it very well behind the red, but they maintain this attenuated response to lipid infusion, whereas lean controls maintain this consistent response to intragastric lipid throughout this time course. In agreement with this and in agreement with the previous data that I showed you, indicating that cholecystokinin is responsible for fat-mediated AGRP neuron inhibition, you see that obese mice have a reduced response to the hormone cholecystokinin after 6 weeks on a high-fat diet, and this reduction in response is maintained even after the mice have lost a significant amount of weight, whereas lean controls, as I showed you before, maintain a really consistent fiber photometry response throughout this time course. So while weight loss restores fast refeeding, and restores the AGRP neuron dynamics associated with fast refeeding, it may not restore the satiating effects of ingested fat. And in fact, we showed, and others have shown as well, that in agreement with this, CCK-induced feeding suppression is actually attenuated in diet-induced obese mice. So in lean control animals, you see this dose-dependent inhibition of feeding caused by CCK administration, whereas in the diet-induced obese mouse, the feeding suppression caused by CCK only occurs at these really super physiologic doses. Unfortunately, we've not been able to do this in mice following weight loss because of some unfortunate mouse sackings that happened. So I've kind of tried to draw a series of conclusions from the high-fat diet studies along the way, and I'm going to assimilate them here as we kind of start to build our model of how diet-induced obesity may alter hypothalamic circuitry driving food intake. So high-fat diet appears to broadly attenuate the response of AGRP neurons to nutrient-related stimuli. Some of these changes appear to be adaptive and would promote weight maintenance, namely the decreased fast refeeding and the decreased anticipatory inhibition of AGRP neurons. Other of these changes could be seen as promoting weight gain, namely the decreased transmission of lipid-mediated gastrointestinal satiation signals. And you could think of these responses as being perhaps maladaptive and promoting subsequent weight gain. These two classes of changes are variably reversible with weight loss. And unfortunately, the adaptive changes recover quickly, meaning that mice become hungry again right after they start losing weight, and their AGRP neurons respond to sensory cues right after they start losing weight. Whereas the changes that are more maladaptive and might promote further weight gain or weight regain tend to persist for at least several weeks following the onset of weight loss. And we think that this is the start of a novel explanation for recidivism in diet-induced obesity, and possibly a neural correlate for the idea of set points, of reset set points. So unanswered questions in future directions, most of them, and the two that we're working on in my lab currently are what are the basic metabolic or hormonal mechanisms of carbohydrate and protein-induced nutrient signaling from the gut to hypothalamus. So going back to the first part of my talk, I don't want to say we've solved how fat causes satiation because I don't really quite believe that to be true, but we're looking for kind of analogous hormonal effects that we see in CCK for fat to what might be seen in protein and carbohydrate-induced satiation. Also focusing on whether absorption is required, whether metabolism is required, and which hormones are, in addition to which hormones are involved. And there's some very cool work on glucose signaling from the gut to the brain from Diego Borquez's group and from Charles Zucker's group, showing that glucose absorption in the intestine is required to transmit this nutritional signal to the brainstem. And we're working on the subsequent step down to the hypothalamus. And then also another important unanswered question that we're just getting started on working on is understanding how obesogenic diets of differing nutrient compositions might differentially modulate hypothalamic dynamics, and what that can tell us about how to nutritionally and potentially pharmacologically approach different types of diet-induced obesity or diet-induced obesity in different classes of patients. And with that, I want to thank the Night Lab. This is where the work that I presented today was done. Zach Knight, as well as Yiming, who's the grad student who did the initial fiber photometry recording from AGRP neurons, and several people in the lab who helped me put together these studies. And then also thank you to the very brave people in my own lab, the first few graduate students who have opted to join me. Here's some contact information if any of you have questions after this is all over, and I'm happy to take questions now as well.
Thank you so much, Dr. Lisa. It was a really interesting talk. Always happy to hear more how AGRP neurons might be regulated or not. It seems like the more we know, the less, well, the more funny and complex the story gets, right? Okay, I think we can we can move on to the questions. We have a few. So I'm gonna start by the ones that are voted. Okay. The first one from Carlos Ribeiro. Do you think that there is a functional relevance for the different mechanisms by which different nutrients act on AGRP neurons? Could there be different effects on other neurons?
I think that there probably is functional relevance to having different mechanisms for different nutrients affecting AGRP neuron activity. Again, it's a little bit hard to foretell what those will be since we really don't know how glucose is doing it. I think on a very short time scale, it might impact food choice, for example. If some of these signals are really rapidly communicated back to the brain, may help you decide if you have a fatty meal versus a very starchy meal in front of you, may help you to decide what to take. And then longer term, the downstream metabolic effects of lipid-rich versus carbohydrate-rich meals are different enough that I can imagine that if not AGRP neurons, then other neurons kind of know what kind of a meal you just ate and know how to, it may help direct subsequent metabolism.
Cool. Okay. The other question we have, do we know, from Eric Meyer, do we know what are the circuits underlying this external mediated regulation of AGRP neurons activity? I guess it's similar.
So if I understand the question correctly, it's what causes the sensory response, like the rapid response to extrasensory signals? Is that the question?
It seems to me. Yeah, it is asking about the circuits underlying this external mediated regulation. So I would guess so, yeah.
Excuse me, sorry, I pulled my nose. Yes, people are working on it, and there are some indications as to what may be driving the kind of rapid sensory mediated AGRP neuron inhibition. There's a population of, I believe, leptin receptor expressing cells in the dorsal medial hypothalamus, which may be contributing to this, and that's from around 2016. It's a few years ago. They showed that there's this projection from DMH that may be involved in this, and then more recently, there was a paper suggesting that projections, I believe, from the BNST may also be involved, and that was a JCI paper from like within the last 6 or 8 months. So people are beginning to work it out, but it's not. It's certainly not 100% known. And I think what's another thing that's not yet known is how sensory regions of the brain, like so like olfaction and visual perception of food may be contributing to that. That's not known yet.
Oh, true. Cool. Okay, another question from Eric Meyer. Did you try a high-fat, high-sugar diet in your nutrients-mediated paradigm? What would be your prediction?
So it's a good question. The diet that we use for the high-fat diet is, as I mentioned, 60% fat, but it's also, I think, 20 something. It's 20 something percent sugar. 20 something percent carbohydrate. So the diet that we're using is not quote unquote low carbohydrate. It's just really enriched for fat. And it still but the excess calories are really primarily coming from fat, and we see this fat specific effect. And what I want to do, what we're working on doing, is rather than using just a fat enriched but not low carb diet, is to use a high fat diet that's low in carbs and a high carb diet that's low in fat, and see how isocaloric over nutrition with those two diets differentially modulates that. And I guess what I would predict would happen, since CCK seems not to be involved at all in the carbohydrate aspect of this, is that axis may wind up being unaffected in carbohydrate-induced obese mice. And I guess I would hypothesize that they develop some kind of resistance to carbohydrate infusion, but I really don't know. We're working on it.
Okay, cool. A question from Anna Fernandes. Do you think that if you used high carbohydrate diet to induce obesity, would mice decrease specifically to glucose?
So that's basically what I was just trying to say. I think that would be my hypothesis, is that a high carbohydrate diet would cause a selective decrease in the response of the intestine or in the gut-brain communication about carbohydrates. But again, that's my hypothesis, but I think little enough is understood about this that I would not be surprised if that winds up being completely wrong.
Cool. Okay, we have a few more. So from Abdul Muqtadir Shafi, water may pass through the gastric gutter, right? Did you control for osmolarity? It's a very technical one.
That's another very, very good question. So I didn't show that data, but we have done the experiments where we do a glucose infusion and then an isoosmolar saline infusion. We've also done this. I've also done this with mannitol, but with saline, which I think is like maybe the cleanest one, with glucose, you see this really robust inhibition. With the same osmolarity of saline, you see maybe a quarter of that inhibition. So I do think tonicity has potentially something to do with it, but it's not. There's no way that osmolarity can account for this whole response. And Zach's lab has also shown that stretch has a lot to do with this. So distension actually within the duodenum is responsible for this, and you can imagine osmolarity having something to do with that because it affects. High osmolar solutions have effects on motility.
Cool. Okay, we have a couple more, if you don't mind, since you still have time. Okay, so from another one from Abdul Shafi, did you measure glycemia of those mice? I think this is IPS or LPS treated 4 hours later. Yes.
Yeah. So we didn't for the LPS experiment. We did not measure.
The LPS through the bacteria membrane. Okay.
Because I guess it certainly glycemia is affected by that. However, what we did do, and I didn't show this. I should have shown this in today's talk. But what we did do, it's in the 2017 paper, is we looked at the comparison between intragastric glucose infusion and intraperitoneal glucose injection. And glucose infusion causes this nice, long, sustained inhibition of AGRP neurons, whereas intraperitoneal injection causes just a small and very transient blip in AGRP neuron activity. So decreases but comes right back to normal. So we don't think that blood glucose is the primary driver of AGRP neuron inhibition. That doesn't address hypoglycemia, but I have other experiments, again, which I didn't show because they're preliminary, but insulin also inhibits AGRP neuron activity. It also causes hypoglycemia, so we're trying to sort out whether it's the hypoglycemia that causes AGRP neuron inhibition or if it's the insulin itself.
Cool. I think I just missed. He had a reply to that comment. So, did you also measure the CCK in these obese mice?
I tried. I didn't get consistent results with the ELISA that I was using, so they're not published. Other people have shown, I think, changes in different directions for CCK in obese mice. Pretty consistently, people have shown CCK resistance at the level of the vagus in obese mice, but in terms of the amount of CCK released into the bloodstream, I think it's a little bit more of an open question, but I think people think it's increased overall.
Cool. Okay, and the last question from Yorick Gitson. Can you please elaborate a bit on the chronobiological aspects of this work, short, immediate versus long-term daily?
Yeah, so we, I guess elaborating it in terms of like the way I think about AGRP neurons and what they're doing, is that we don't feel hunger and satiety over the time course of a single meal. And the analogy that I use a lot of times to talk about this is, you can imagine when the pandemic is over, you go on a really nice long vacation with your family, and you eat at restaurants every day, and you eat rich meals every day with your family. And then after a week of eating really rich meals, you come home and you kind of just want salad, or you want things that are very light, and you tend to not be that hungry. And this has been done formally. I stopped presenting these because they're like actually really horribly unethical experiments from back in the 60s, where prisoners actually were force fed for like 12,000 calories a day. They were forced to gain 20-30% of their body weight in a pretty short time period. And when they went back to being ad libitum fed, or able to control their own food intake, they almost all went back, had a period of profound hypophagia, and then went back to normal feeding. So we regulate our appetite over multiple time scales. And additionally, I think our appetite as people is regulated by what's available too, right? Like, I'm not hungry right now sitting here talking to you because I gave a talk, so there's a little bit of adrenaline. I drank some coffee, so my appetite is maybe a little bit suppressed. But if somebody placed a donut on my desk over there, I would suddenly be very hungry. And I think AGRP neurons are the neurons that are helping to integrate all of this information. So is there a donut in my environment? Have I eaten recently? And has my food intake over the last, period of days or weeks been abnormally high or abnormally low? And so that's how I think about the time scales, is that we're not hungry on a single time scale, where our hunger is controlled by our nutritional state over multiple different time scales.
Okay, cool. This is a really, really nice answer. I think it really nicely wraps up your talk. So thank you so much for being here with us today, for giving me a very interesting talk. I would like to thank everyone also who participated. And you can also join us next week. We will be hearing about Dr. Lisa Stowers on leveraging olfaction to understand how the brain and the body generate social behavior. So with this, I'd like to thank you, and we will now move on to the Q&A session, in a couple of minutes for you to have a break. Okay. Thank you so much, everyone.
Bye.
Bye-bye.
Thanks, everyone.